Was. Hello, a very warm welcome back to the session, the second session of the pharmacovigilance. I must hand over Dr. Shikha to continue with the session from the next part of the session. So Dr. Shikha, are you hearing me? Dr. Shikha.
Hello. Yes, sir.
Uh, Please continue the session.
Okay. A very good afternoon to everybody. So myself, Dr. Shikha. So we'll proceed to the history of pharmacovigilance. So we were doing Stanford prison experiment. Earlier we discussed the various experiments which were performed during the World War or during any kind of researches. So which were involved in the pharmacovigilance and in the study of various products or various medications. So now we are going to start next experiment, that is Stanford prison experiment. So next slide, please. So this is a psychological experimentation of assessing the effects on becoming a prisoner and a guard. The volunteers were college student, that was 24 selected from 75 respondents. Means they have taken the consent from 75%, but only 24 were selected. Next is the site for that presentation was Jordan Hall basement, Sanford Psychology building. Next is investigators, Professor Philip. Please, mouse, please say it. Next is the site was Jordan Hall basement, and next investigators were professors of Philip Zimbardo and his research team. The funding agency was U.S. Office of Naval Research. Next, the duration of that study was August 14, 1971. Planned for 2 weeks. It was basically planned for 2 weeks. Now, the conclusion of that research was that the participant acted differently than normal. Study was questioned to be unethical. They said that this study is not ethical, not valid. So that's why. Next slide, please. So these are the monster study. Next one. This is the photograph of that. So yeah. Next. Speech therapy experiment. There was an experiment which was performed on the speech therapy. So there were volunteers, 22 orphan children, 10 actual stirrers and 12 normal. So there is a difference. Some were 10 students were, sorry, 10 orphan children were stutters and 12 were normal. The site was Davenport, Iowa. Investigators were Mary Tudor, a graduate student supervised by Wendell Johnson, professor at University of Iowa. So duration of that study was January to May 1939. So next outcome was the children receiving negative speech therapy, originally normal, then suffered negative psychological effect and some retained speech problem for the rest of their lives. Next is ethical issues were questionable as children suffered emotional setback and were compensed at 2000 in 2007. Next, please. Next. So this is about project 4, study about effect on health of people after radioactive fallout having high yield. Primary cause is radioactive yield from Castle Bravo nuclear test. At Bikini Atoll on March 1, 1954, the population that was in this study that was resistant of Marshall Island or US, and period is about primary study 75 days after the exposure, follow-ups at 6th and 12th month post exposure. After the exposure, the primary study was taken, or primary follow-up was taken on 6th and 12th month of the after the study. Next is investigators were department Department of Energy Committee, Eugene P. Krotikite, Project Officer. So, outcome of this study was post 5 year exposure, rate of stillbirths and miscarriages doubled. Further decades, thyroid carcinoma in children's, and by 1974, one third of exposed development neoplasm. Susception, that was Marshallese alleged that the donation was pre-mediated, treatment provided was not up to the mark. Means they said that the treatment which was provided, it was not up to the mark, it was not valid. Next slide. So this is Tuskegee's syphilis study. So syphilis is basically a disease which is caused by it is basically a STD, that is sexually transmitted disease. So next, the study about leaving syphilis untreated and investigation of progression of disease. So basically, the person who were having already diagnosed with syphilis, they were left untreated. On that patient, or that patient, the study was done. So in this, the population was, which was considered for the study, that was 3399 syphilis infected African American male sharecroppers plus 201 in control. Next is site Tuskegee's, Alabama. Alabama. Investigators were used, that is US Public Health Service plus Tuskegee's University. Next, the duration of the study was 1932 to 1972. Next is outcome. Only 74 men survived. Out of this much of population, only 74 men survived. 28 died originally of syphilis. Some 100 died of related complication. 40 of the other wives contracted syphilis. 19 of their children were born with congenital syphilis. Next, uh, ethical concern. Despite the authorism of penicillin in 1947 as safe and standard treatment of syphilis, the subject was denied for of the treatment. Next slide. So this is Nazi's experiment. So we'll discuss this again. Next one. The wide series of medical experiment on various disease and procedures, bone, muscle and nerve transplantation experiment, sulfonamide experiment, dairy bomb experiment, experiment with prison, etc. So earlier we discussed these studies earlier also. So in this study, they are saying study population was prisoners, a very large number in number, including children. Means children were also involved in this study. The investigators were Nazi Germany physician. Next is site was concentration camps. Period was early 1940s during the World War II and the Holocaust. Outcomes were the outcomes of this experiment resulted in death, trauma, disfigurement, or permanent disability. So ethical concerns are nothing was ethical. They said that the study is not ethical at all. So next, outcomes were next is the list of experiment carried out by the Nazis. So that is experiment on twins. So this is basically the list which the Nazis carry the experiment. So these are the list of that experiments. So that is experiment on twins, then bone, muscle and nerve transplantation experiment. Next is head injury, experiments, freezing experiments, then malaria experiment, immunization experiment, mustard gas experiment, sulfonamide experiments, seawater experiment. Next one is sterilization experiment. Next is experiment with poison. Next is incendiary bomb experiment. Next one, high altitude experiment. Next one is blood coagulation experiment. So these were the experiments which were carried out by the Nazis. Next one. So, high altitude experiment. In early 1947, prisoners at Dachu concentration camp were used by Sigmund Rascher in experiments to aid German pilots who had to eject at high altitudes. Means some of the pilots were there who were allowed to go at high altitudes. A low pressure chamber containing these prisoners were used to stimulate conditions at altitudes up to 20,000 meter, that is 66,000 feet. It is rumored that Rasher performed vivisections on the brains of victims who survived the initial experiments. Means when they were sent to high altitude, the person who survived there, the pilot who survived there, the next experiment was performed on their brain. Means they were taken as a victim for the performing of next experiment. Next is, of the 200 subject, 80 died outright and the others were executed. Means 200 subjects were there, 20 died and others were subjected or executed. Next one. So next was blood coagulation experiment. Sigmund Rascher experimented with the effect of polygal, a substance made from beet and apple pectin, which aided blood coagulation. Means these are the factors which are helping in uh, blood coagulation. So he predicted that the preventive use of polygal tablet would reduce bleeding from the gunshot wound sustained during the combat or during surgery. Subjects were given a polygal tablet and shot through the neck or chest or their limbs, amputated without anesthesia. So Rasher published an article on his experience of using polygal without detailing the nature of the human trial, and also set a company to manufacture the substance staffed by the prisoners. Next one. So, next is the history, that is, morphine was isolated from raw opium in 1805 by a German pharmacologist, Friedrich Wilhelm Adam Suttner. So, morphine is a potent suppressor of pain and is a very useful drug in painful condition, especially a severe chest pain arising due to heart attacks. So, it is also used induces sleep in no time. So basically, they are talking about morphine, which was discovered or which was isolated from opium in 1805 by a German pharmacologist. So they are saying that morphine is a very potent suppressor. It is basically widely used for very severe pain, chest pain, in many of the hospitals during so many treatment. Next. Friedrich William Adams Turner. This is the picture, and this is the structure of opium and morphine. Next, this is Joseph Pelletier and Joseph Beneme. So these are basically different structures. Next slide. Next one is John Stith. Uh, Next one, addiction to invention. So they this he is the person who discovered Coca-Cola basically. Next is soldier in American Civil War. In April 1865, he got wounded in chest during Battle of Columbus. Means 1865. May he was get he was wounded, means he was injured in a chest during a battle of Columbus. Next, they prescribed morphine to him, so it became addicted to morphine. So started searching cure of the addiction, performed experiments on herbs, that is coca leaves, made a beverage, that is coca leaves plus wine plus cola nuts, named Pambetron French wine cola. In 1886, alcohol prohibited in Atlanta, then country Georgia, replaced wine with sugary syrup named Coca-Cola. So that was a time when Coca-Cola was discovered by John Seth. Next slide. In 1944, multi-center studies were performed, that was conducting large trials at multiple sites represented a significant change in how studies were conducted. For the first time, trial were conducted at different sites using the same protocol with all the centers result assessed together. Now they are saying कि, conducting large trial. They were conducting a very large trial, and at the different sites. The protocol was same, but they were doing that trial on the different sites using same protocol with the all the centers, and the result was assessed together of different sites. The result was assessed together. This finally allowed for larger members of participants and a wider range of population group to be studied with which strengthened research trials, designs and analysis. So next is today most large trials are conducted at multiple clinical centers, research centers. Our monitors at uh, IMARC can attest to this, as the majority of our monitor studies take place at numerous sites across the country. Means a large number of studies are conducted at multiple clinical sites, and they are monitored by IMARC, and the result can be attested. Next one. In 1944 to 1974, a human radiation experiment was performed. During the Cold War, thousands of US citizens became the innocent and unknowing victim of over 4,000 secret experiments sponsored by the US government aimed at determining the effect of atomic radiation on the human body, often using doses likely to harm the subjects. Means they were during the World War or Cold War, you can say, the US citizens became an innocent victim to perform these experiments. They were the subjects, but they don't know that they are the subjects of some of the experiments. So they were giving, the US parties were giving them a very high dose that can harm their body. So the majority of victims included vulnerable groups such as prisoners, pregnant women, children, and impoverished and disabled patients. Next slide. Next is 1947 Nuremberg Code. During the Nuremberg trial, 23 members of German Nazi Party were tried for crime against humanity for the atrocious experiments conducted on unwilling prisoner of the war. Means they were not interested and they were not willing to be the participant, or they were not interested to take part in this research, but they were forced to do that. The resulting verdict contained a set of 10 ethical principles of human experimentation. This code established the requirement for informed consent, absence of coercion, properly formulated scientific experimentation, and benefits towards experiment participants. So, next slide. In 1951, Henrietta Lacks. So, Henrietta Lacks' contribution to medical science is enormous, yet she never knew or intended it to happen. She was unaware of this. During treatment of cervical cancer cells, cells were taken from her tumor without her knowledge. Researcher George Otto Gay took these cells and noticed that remarkably the cells could be kept alive in a culture. Her cell would eventually become the Hela, he for Henrietta and La for Lax. Immortal cell line, commonly used in biomedical research. Since the extraction of Henrietta's cell, some 20 tons of cell have been grown for the Hela line and distributed to the researcher according to the world. These cells were used to test the first polio vaccine in the 1950s and have since been used for AIDS and cancer research as well as many other scientific researches. Next. 1967 Declaration of Helsinki. The declaration expands on 10 principles necessary for ethical human experimentation set forth in the Nuremberg Code. Development by the World Medical Association, the Declaration of Helsinki is generally regarded as the cornerstone document of human research ethics. Since its inception in June 1964, it has undergone six revisions, the most recent coming in 2008. The focus of the declaration is on respect of the subject's right to self-determination and their right to make informed decision regarding research participation. It stands as a clear and powerful statement that the right of the human subject shall never be compromised for sake of science. So in this time they made it compulsory that the subject should has each and every right, means they should they should be told that what is going to happen and what is to be done with them. And basically it was very very important for the subjects to know this. So they already said okay, the health of the subject should not be compromised for the sake of science. For any discovery, the health of the subject should not be compromised. Next slide. So, 1974, the National Research Act. The US President Richard Nixon signed the National Research Act into law in response primarily to the Tuskegee syphilis study. The act required that all research using human subject be reviewed by an institutional review board as another step to ensure the protection of human subjects. Means they said that the researches or the experiments which are performed earlier, so which was not ethically done, so they were saying it to review. Basically again they were saying to review it, and they were saying to perform that experiments again with the proper protocol. Next. So 1974, that was FDA Bureau of Medical Devices and Diagnostic Products. Advance in technology led to a significant growth in the medical device field, yet the industry was still operating based on device regulation from the Federal Food, Drug and Cosmetic Act of 1938. Clearly updated regulations were needed. So, in February 1974, the FDA created the Bureau of Medical Devices and Diagnostic Products to meet this need. Next. Next one is 1979, the Belmont Report. So the development developed primarily in response to the Tuskegee syphilis study. The Belmont report explains the three unifying ethical principle to guide human research, means respect for the person, benefices and justice. So these were the 3 points which were very very important or ethical principles for the human research. That is, they were saying first is respect for the person, benefices and justice. The third was the Belmont. Next one is the Belmont report has become the primarily ethical framework for protecting human research subject in the United States. So next slide. Next one is 1981 FDA regulation title 21. So in follow up to the Belmont report, FDA and the Department of Health and Human Services formally revised regulation for human subject protections by creating title 21. which includes regulation for the protection of human studies, Part 50, financial disclosure, Part 54, IRBs, Part 56, investigational new drug application, Part 3, 312, next one, investigational device exemption, Part 812, and electronic records. That is part 11. So, FDA regulation title 1. So, this was basically the rules and regulations for the protection of human subjects. Basically, in this, many of the parts are there in which there are different ethical things for the safety of the subjects involved in the clinical research. So basically it was firstly human subjects, protection of the human subject. Next one was financial disclosure, means if any project is participating, any subject, sorry, is participating in that project, so they were getting financial help also. Okay, so next is investigational new drug application. Means if you are adding any kind of new drug or researching any kind of new drug, so that was also placed in this article. So next, next one is 1990, that is International Conference on Harmonization Guidelines. ICH, this is very, very important. So, European nation along with Japan and United States begin creating plans for global harmonization of regulatory requirements to reduce duplicate, time consuming, and expensive procedure needed to market products internationally while maintaining safeguards on quality, safety, and efficacy. This need led to the creation of ICH guidelines in April 1990. So basically, it was a regulatory requirement. So that's why to reduce the duplicate or time consuming and expensive procedures to internationally market your product. So, due to that, international conference on harmonization was uh, introduced in April 1990. So, next, in 1991, the common rule is yet another set of ethics involving the protection of human subjects. Means this is in 1991, the common rule. This was also related to the protection of human subjects. So, captured in the Code of Federal Regulation, Title 45 and Part 46, the rule describes the type of research subject to regulation. The rule also set forth requirement for the institutional review boards oversight of clinical trials and details protecting or protection of human subjects regarding informed consent. Means is at this time they were providing a informed consent to the participants or to the subjects. Basically subject is the person who is involved in the clinical trials. So they are saying that they were providing a informed consent through this rule. It was made compulsory to provide the subjects a consent in which it was clearly written why the study is performed and what to be given, what compensation is to be given to the uh, subject if any mishappening happens. Next. So 1993, MedWatch. MedWatch was launched by the FDA, that is Food and Drug Administration, as a system designed to collect health professionals report of adverse events involving medical products. Next is when the safety hazards are detected, the FDA issues medical product safety alerts or order product recalls, withdrawals, or labeling changes to protect the public health. Next, next is 1996, the World Health Organization or ICH guidelines for good clinical practices. The guidelines addresses justification for the trial and protocol, protection of the subject, responsibilities of the investors. Basically, the World Health Organization or the ICH guidelines for good clinical practices means clinical trials. Whatever you are making for the use of human beings. So that was these were some rules. The first was the guidelines addresses means in this guidelines it included justification for the trial and the protocol, ki why you have used this trial, why you want to perform the trial, and proper defined protocol. Next one is protection of subjects. Basically, you ensure the protection of the subjects who are involved in this clinical research. Next is responsibilities of the investigators. The investigators who were producing or who were carrying out this type of studies, so the responsibilities of that investigators were also mentioned. Sponsors and monitors. The person who is sponsoring your clinical trial, he or she was also mentioned. Then monitors also. Next one, the assurance of data integrity and product accountability. Means your data is uh, proper integral. Next one is the role and the roles of regulatory authorities. So different regulatory authorities for checking the validation of the project which was going on, which was carried out. Next, so this is basically the timeline of clinical research. The whatever we did earlier, it is basically in a tabular form. So first is 605 BC. That was the Book of Daniel. 500 BC was the Hippocratic Oath. Next is 1025 AD was Avicenna Canon of Medicine. 1537, that was first clinical trial of novel therapy, that was embryois pair. Next is 1747, James Lind's Scurvy's experiment. Next was 1800, arrival of placebo. Then 1887, National Institutes of Health, that is NIH, founded. Next, 1908, FDA Pure Food and Drug Act was established at this time. Next is 1928, Sir Alexander Fleming discovers penicillin. Next is 1932 to 1972, the Tuskegee's syphilis study was performed. Next was 1937 elixir sulfanilamide disaster. Next is 1938 Federal Food and Drug Cosmetic Act A series. Next is 1939 and 1945 World War II experiments. Next slide, please. In continuation, in 1944, multi-center studies were performed. Then 1944 to 1974, human radiation experiments were performed. After that, 1947, Nuremberg's code was done. Then after that, 1951, Henrietta's lax was done. After that, 1974, the National Research Act was established. Then again in 1974, FDA Bureau of Medical Devices and Diagnostic Product was established. Next is 1979, the Belmont Report was established. Then 1981, FDA regulation title 1, this is very important. So FDA regulation title 1, this is very important. Okay, next is 1990, International Conference on Harmonization Guidelines, ICH, this is again very important. Next one is 1991, the Common Rule. And next is 1993, MedWatch. 1996, the World Health Organization guidelines for good clinical practices. So with this, we are over with the history of pharmacovigilance. So this was all about the history of pharmacovigilance. Now, I hope you understood everything, or if so, now I forward the the session to my colleague, Mr. Hardik. So, Mr. Hardik, you can start the session now. Thank you so much.
Thank you, ma'am. I will continue the session. There is the global pharma market was worth of dollar 934.8 billion in 2017, and it will reach to it has already reached to 170 billions in 2021. The estimate is in 2025 that it will grow more than the 7%. The next slide, these are the various types of departments available in the hospital. These are the various departments available in the hospital on the basis of anatomy, cardiology, endocrinology, forensic medicine, neurology, orthopedics, pathology, psychiatric, pneumatology, biochemistry, emergency, examinations, hematology, nephrology, ophthalmology, oncology, microbiology, and the surgery departments. These are the various departments available in the hospital. There is a branch of a study of adverse drug reaction is called pharmacovigilance. This is the simplest way of the definition of a pharmacovigilance, the branch of study of adverse drug reactions. It means there is an unwanted effect or an unwanted reaction occurs in the body due to the treatment. Okay, here are some examples. So the drugs are safe or not. See the photosensitivity from amiodarone, the severe extemia of amiodarone infusions, NSAIDs induced peptic ulcer, goiter, hypothyroidism, secondary of amiodarone, bleeding due to anticoagulants, exothermal rashes from penicillin in patients with the previous known allergy or the area. The next slide, these are the some regulatory bodies, those who are having a pharmaceutical regulatory bodies. So we will discuss one by one. Uh, who regulates drug safety? First is Slovakia State Institute for Drug Control, section of drug safety and clinical trials. Czech Republic State Institute for the Drug Control and Pharmacovigilance Department. In UK, medicine and healthcare product regulatory agencies and vigilance risk management of the medicines. In the USA, Food and Drug Administration Centers for the drug evaluation and the research. So in India, it is a Central Drug Standard Control Organization, which is also known as a DISCO. And nowadays, the Pharmacopoeia Commission is also regulating the PVPI centers. The next slide, this is again in Europe, there is a European Medicine Agency. In USA, there is a Food and Drug Administration. In United Kingdom, there is a Medicine and Healthcare Products Regulatory Agencies. In Australia, there is a Therapeutic Good Administrations. In Canada, it is known as Health Canada. In China, it is known as State Food and Drug Administration. In Denmark, it is known as Danish Medicine Agency. In Switzerland, Swissmix, Swiss Agency for Therapeutic Products. In Japan, Ministry of Health, Labor and Welfare. In Germany, Federal Institute of Drugs and Medical Devices. In Brazil, it is an Agência Nacional de Vigilância Sanitária. In New Zealand, that is MedSafe, Medicine and Medical Device Safety Authority. In Sweden, it is a Medical Products Agency. In Netherlands, it is a Medicine Evaluation Board. In Ireland, that is a Irish Medicine Board. In Italy, it is an Italian Pharmaceutical Agency. In South Africa, Medicine Control Council. In Singapore, Center of Pharmaceutical Administratives Health Science Authorities. In Thailand, it is a Ministry of Public Health. So these all are the agencies in the different countries that are available in there. See this. This is a diagram in which medical input is equal to medical output. There is a bridge built by the pharmacist. That means the pharmacists are dependent on the both medical inputs and the medical outputs. The picture, that is a drug safety and prescribing association relationships. The both are fighting with each other. It means there is a drug safety parameter also and the prescriber associated relationship also. In the next slide, the drug safety associates. It looks special, but actually not. In the photograph, the picture is looking very special, but there might be an drug interaction, or there might be an food interaction between the all. The next, what is the prescriber? Looks simple but special, but having the specialty. See, there is a picture having a place. The specialty organizations are having the specials. In this pharmacovigilance, the teamwork is very necessary. The teamwork is in between the prescribers, drug safety associates, nurses and sporting staffs, pharmacists, patient, their relatives, or any common person. There is a teamwork between the whole of the families. In the next slide, the Murky Koffler, founder of Shopper Drug Mart and co-founder of the Four Season Hotels. The next slide, yeah. Then is a thalidomide disaster. That was a wonder disaster. The thalidomide drug was used for the pregnancy, in which the result came that there are so many babies are been having without their limbs. That is a baby without limbs. As in a history of a thalidomide disaster between 1957 to 62, thalidomide tragedy, the greatest of all drug disasters, thalidomide has been introduced and welcomed as a safe and effective hypnotic and emetic. It rapidly become popular for a woman for the treatment of a nausea and vomiting in the early pregnancy. But tragically, there are the thousands of children which bones without their limbs. The in the next slide, I must show you the news. This was the Francis Onion Kinley drug defective news, and the museum we are having in the USA, that is a thalidomide museum. These are some pictures of the thalidomide museum. See there. See in this, they have preserved the babies. Those who are born without limbs. Some are not having their eyes. Some are not having their legs. Some are not having their arms, or some are having their any part of the body. Gradually, thalidomide was retinoduced for the treatment of few skin diseases, including leprosis, erythema nodum, serves muscular ulcer. For example, associated with HIV infection and back disease. See here. For the recovery of that, there was an artificial arms were built, pair of artificial arms for the child, Riopotam, England, in 1964 after the thalidomide tragedy. But there was so many chances of the devices that can produce the shock to the babies also. So it might lead to the serious conditions like having a patient admitted to the ICUs or a deaths. In the next slide, these are the some examples like the drug disasters, Merrick for the human drug turned nightmare. For Fen-Phen, Des, Brackley, Vabox, Bedax, Resolin, Sedanin, Pico, PPA. The composition was the fenfluramine and penthamine. There was a weight loss. It is used for the condition of a weight loss, but it damages the heart valves in 1997. Then diethyl subustyrol, it was used to prevent miscarriages, but the tumor that kept appearing in the daughters of the woman who had taken this. The cystatin treatment, it is used for the treatment of high cholesterol, but there was the adverse effect like severe muscle disorder known as rheumatophysis, which clogs the kidneys and with protein from dying muscle tissues. In the next slide, there are some more examples like premolin is used for the Abbott laboratories. It's used for the central nervous stimulator, but it produces the liver toxicity. In esthumazide, it is used for antihistamines, but it slows down the potassium channel of the heart. In the next slide, there is a tafnidine that is used for antihistamine, but the adverse effect like causing the life-threatening heart problems when taken in the combination with other drugs, specifically in erythromycins. And this is the all about the some examples. In the next slide, there is a drug related to the safety problem since 1961. In the year that polydexide in 1974 safety corner due to interpretates. These are some or more examples. The next slide, the withdrawal from the market. If any drug is causing more than the 50% of the ADRs, the company or the US FDA or the Sadisco mention the notice to the company, please withdraw this medicine from the market, and they issue a notice to the market that do not use this drug as per these indications. Next, the pharmacovigilance in India. The pharmacovigilance in India, the world's largest democracy population, more than 14 million peoples. The age distribution is less than 14 years is equal to 30%, and 15 to 64 years, that is a 60%, and more than 65% is a 7%. This is an estimate chart of the population, that is the age distributed. In the next slide, advantages in India. There is a large population, potentially large world scale area database can be more. So that in India there is a treatment facilities are more. There are having lot of a disease. For example, we will talk about there are so many patients of cancer, neurology, alcoholic liver disease, diabetes, hypertension. Well, what are the sources? The sources and the pharmacovigilance services. The sources might be the clinical trials going on, post-marketing surveillance, the SADISCO, the FDA, they daily monitor the adverse drug reactions that has been reported by any hospital or any AMC center nearby them. They do the audits of that report, and the annual summary was prepared, and the services are in the healthcare professional letters, public advisories, product risk assessments and mitigation strategies, risk management plans, and regulatory inspection supports. This is a chart. The basic process of automation. The next is robotic process automation. Then the third is cognitive automation. And the last is artificial intelligence. Nowadays, there is a lot of use of artificial intelligence. For example, we want to know anything. You just ask to AI, just I want to know about the pharmacovigilance. He will provide you the data, but in a limited access that you have to verify from the sources like literatures, any other course materials, or any some websites like Google Scholar, PubMed, elsewhere, Scopus directory, Web of Science directory. You have to follow that type of sources to identify or an authentic areas. This in this picture, it is the man holding a balance in which there are some benefits, in which there are some risks, that they are equally measured during the time. This is the process in which the data is ADR reporting, active surveillance, past media, network monitoring. Then there is a knowledge about safety signaling, publications, media, and network informations, regulations like regulatory actions, risk assessment, usage, appropriate use of medicines, public health. In the public health, there is a maximizing benefits, missing risk. In the healthcare, there are changes of behavior, changes in clinical practices, patients change behavior. There is a term called good clinical practices that comes in the hospital. Why? Because if any new drug has been launched in the market, it has been passed the preclinical, clinical trials. Then it comes to the post-marketing surveillance. In the post-marketing surveillance, also doctor monitors that drug reactions. What the reactions happening to the patient? For example, if somebody is ill, I'm providing. If I'm having a fever, so I will take some analgesics or any some. If I'm having pain, I will take some painkillers. But it is going towards the positive feedback or a negative feedback. If I have cured my disease or I have treated that disease, so it is having a positive feedback. But if it is causing an other reaction rather than going to the specific targets of our body, that means there is some problem in the reaction in the body with the drug, or there might be some adverse drug reactions, or there might be some adverse drug effects in the body. So pharmacovigilance is having acting to protect public health, in which we measures patient safety, education and training, public health, rational use of drugs, WHO and Uppsala monitoring centers, pharmaceutical companies, hospitals, academia, patient, healthcare workers, national and regional pharmacovigilance centers. The next slide, what does the pharmacovigilance do? That is the monitor, code, assess and understand, report and communicate. In this, there is a monitoring of a patient. Those are under the treatment, or those who are taking a self medication. First of all, we have to stop the self medications. The self medications might be the major having the impact in the adverse drug reactions, because everybody knows that this is a painkiller, we can take that. But if I am having a severe headache from 3 days, I must visit a physician or a doctor to take his recommendation. It might be another problem also in our. And this is an overview of pharmacovigilance. There is a spontaneous reporting, literature reports, clinical trial data, regulatory reports, licensing partners. They are the measured by the database, then data review, then it is circulated all over the world. There might be the submissions, there might be the amendment prescribing informations, there might be the review of marketing status, inquiry responses, worldwide regulatory reports, or other licensing partners. The next slide. These are the services provided that ensure safety and expedited approvals. See, it is a multiple tasking things. The pharmacovigilance has to communicate with the clinical, medical affairs, and local authorities. As well as they have a safety narrative writings, the control safety, surveillances, safety signal managements, aggregates report management, adverse event and product complaint managements, individual care reporting, cell center functions, safety database services. If I talk about the pharmacovigilance, it is a vast and wide course. There are so many parameters to learn. There are so many literatures we have to find. We have to get an update about recent activities, the recent approval of drugs, recent clinical trials, the recent notifications about the drug, that the safety for the safety parameters. This is a circular providing product services for them, the development to the market ensure patient safety. First is full clinical or PVPI medical monitoring support. Second is case processing and literature surveillance. Then third is safety writing services. Fourth is data safety monitoring record. Fifth is event adjustment. Sixth is signal detection. Seventh is qualified persons responsible for pharmacovigilance. It means if you are having a knowledge about the clinical studies, if you are linked with the healthcare professions, if you are a non-healthcare profession also, you can also link with the healthcare professions. You can learn about the how to search for the literature, how to write the literature, how to make a file, how to prepare the proper file of adverse drug reaction, how to write the safety services, how to monitor that, and the event adjoint. What is the signal detected? Then the pharmacovigilance is having a patient, policymakers, that is our regulators, physician and medical regulators, pharmaceutical industries and associations, public and press and media. In the next, it is the global market insights. This is all about 2018 to 2025. That there is a more than 4.3 billion, then there is a 10.6% increase, that is a 8.9 billion it up to 2025. The preclinical studies arrangement segments, contact outsourcing segments, and phase 1 clinical trial segments. There was an regional outlook, that is the APAC market value by 2025, that has been already reached. Then the marketed CAGR. See, this is a teamwork application which we knows about the expert team, MR team, executives, medical advisories, risk mitigation, communication, patient, doctor. This is all the process that there is a team in the members of the team who works in this. And then is another example. FDA recall 337 drugs in 2014. The data increases day by day, but if I talk about the previous data or the history of the data, in 2012 there were 291 drugs, 315 drugs in 2013, and in 2014 there was a 333 drugs that has been recalled from the market to stop that production because they are causing so many adverse drug reactions. For examples of licensing drug withdrawn after the marketing of the drug safety reasons, first is thalidomide, second is paracatol, third is fenformin, fourth is ribofox effects, fifth is valproate depression, movement disorders, and sixth is thalazine. So vigilance always works towards the drug safety. What is the aim and scope of the pharmacovigilance? To improve patient care and safety in relation to medication or and all medical or paramedical interventions. To improve public health and safety in relation to the use of medicine. To contribute the assessments of benefit, harms, effectiveness, and risk medicines. To promote understanding, clinical training, and effective communications to the health professionals and the public. Why do we need pharmacovigilance? It is for humanitarian concern. ADR may cause sudden deaths, promoting rational use of medicines and adherence, and the ethics to know of something that is harmful to the another person who does not know and not telling it's unethical. The minimum requirements for the functional pharmacovigilance system, that is a national pharmacovigilance center with with designated staff, at least one full-time stable basic fundings, clear mandates, well-defined structure and roles, and collaborating with the World Health Organization programs for international drug monitoring. And the second step, the existence of national spontaneous reporting system with the national individual case safety report, that is called ICSRs, from is that ADR reporting form. In the third slide, a national database or a system for controlling and managing ADR reports. And the fourth point is a national ADR and pharmacovigilance advisory committee able to provide technical assistance on casualty assessments. In the previous area in India, there was the Naranjo scale was used, but nowadays the World Health Organization UMC scale is used to assess the casualty assessments. The clear communication strategies for routine communication and crisis communication. For any information on a ADR or a lack of efficacy connected with the use of medical devices or drug, the in ADR occurring this is in a course of the use of a drug from drug and overdose, abuse, misuse, non-appropriated use, withdrawal, nursing, during healthcare, even if no ADR has been observed from the overdose, whether accidental or intentionally. Then there is a regulatory authority which regulates in the communication and risk mitigations. How PV workflow? First is data collections, that is the ICSRs has been collected from the various hospitals or any AMC centers. Data entry is to be in the database. Nowadays, the pharmacovigilance program is using a database like VigiFlow. That is the database in which the AMC centers report the ADRs. Then there is a review panel by the NCC casualty assessment. Then there is a signal detection or a UMC Sweden. Then the signal detection comes under the aggregate reporting, and then it comes under the regulatory authorities of SADISCO. Then the action is been taken. So this is a program communication, that is a SADISCO headquarter, all the training centers and all. So this is all about the today's session. I would like to inform to the participants that please be attentive in the sessions and please mark their attendance. Hope you all are doing well. And so once again, the warm greetings from the School of Pharmaceutical Sciences. That's all for the today's session. The two sessions will be conducted in every Saturday of every week. The session link will be sent before the session into your WhatsApp group that you have already entered or has joined in the group. So thank you everybody for your presence. Once again, the warm greeting from the School of Pharmaceutical Sciences, City University, Ludhiana. Thank you everybody. Gandhi sir. Okay. गाइड तो छुट्टी ते आया इतनी बंदी है टेबल होंगे है नहीं भाई सारे लॉग इन करके बंद कोई नहीं कर रहा
ये टाइम बहुत लगे थे
रहने दे कोई
कब
गल
ਨੂੰ ਕਰਾਂ
कल
या मंडे होना ना
मंडे में छुट्टी थे मैं तो ब्याह पे चलना है
अच्छा
ट्यूसडे से
चलो ट्यूसडे सुबह ले आऊंगा मैं